Scott's just doing that, trying to divide us.
Sorry, Chris. I was actually being honest for science.
Oh, fine.
3-0. Where's my paper? I gotta write this down.
I don't think I saw that one.
So the dead sponge is the yellower part, and then the only remaining live part is the sort of crystalline white part. And after not having seen the sponge for something like 20 years, we've seen it on the last 2 dives in multiple colonies. This is,
let's come a little wider.
Again, really amazing.
Not full, just wider.
Well, that's exactly why I brought this up, independent of your internal competition there. Is that it's funny when we see these patterns, and it shows how much we have left to explore. Some things we think are so rare may not be as rare once we get down here and have a good look.
Yeah, it's definitely there's patchiness in the deep sea, and you just got to be at the right spot if you're looking for a particular animal, because you can have one animal on one side of the seamount. It doesn't exist on the other side of the seamount. It could be on one seamount and not on the next seamount over. So you just never know. Every time you go down, you just never know what you're going to see. And I'm starting to wonder if we're learning what we're not going to see. They'll turn up. I'm sure they will.
They'll turn up. Yeah, but see, these crinoids. I don't know if they're the same species, but I think they're doing pretty well in this area because they all are quite large.
Yeah. This guy, I'm sure, is in the Okeanos online guide here. And I think it is. I'm trying to look on the guide.
These are those purple ones with the yellow cirri.
Yeah, with the yellow cirri, that's it.
Yeah, what is that?
I have a feeling it's going to be Comatulida or something like this as far as we were able to get on it.
There are also zoanthids overgrowing part of the branches on the left side of this coral.
Oh, yeah. So zoanthids are sea anemone-like animals, and they seem to have a propensity for overgrowing other colonial animals.
So just a little heads up for those of you watching. Dave, the pilot, already mentioned, and it's sort of a reminder that while the ROV is fixed, it has a little bit of a wobbly wheel. So he's being extra cautious here approaching this type of terrain. And so we're going to, approach things a little bit slower until they make sure everything is working 100% again. So this is a pink coral in the Coralliid family, and again, I don't know what the identification of that crinoid is. We don't have a good one except that it's a feather star, and that's it.
There's 2 animals here. There's a base there. There's a shrimp I couldn't get to.
Well, it could be. So in the Hurl Gallery, this is some feedback from Chuck Messing.
Oh, a thalassometra.
Yeah. Does it have enough arms to be a thalassometra, this one here?
Possibly, yeah. It certainly has the yellow cirri. That discordant coloration.
Does it have prehensile cirri? I wasn't even paying attention. I wonder if the pilot could...
I think so. It's adhered to the surface.
What are we looking for, Chris's?
The feet on this particular black crinoid that's just a little bit up further.
Oh, yeah.
We didn't notice.
Yeah.
It does. It has prehensile.
Yeah.
So maybe Thalassometridae,
the
family.
A lot of the taxa that we're looking at often need to be examined very carefully in order to come to a correct identification. And sometimes in spite of all the wonders that our video brings to the detail. There's nothing that's like just being able to sort of look at the fine skeletal articulations and other sort of subtle characters that are on the animals. And sometimes animals are prioritized for collection in order so that we can figure out what species they are.
Notice the tether blowing pretty straight back. I don't know if you feel
I
any
think we're
current,
good.
but
We can move on.
We can come wide, please. I don't see the crinoid waving around much there.
it's true.
And I haven't felt any
Another
forces to-
primnoid, Narella on
curious
the
what-
left, and in the
look like it was
background, where they got good images of
blown.
those. So that wall definitely had the most animals on it, and then we've still got a few on the sort of flatter terrain.
We have more up in our future here.
A lot more up.
Yeah, we're going to try to stick to the edge of this landslide escarpment too, which we expect to
Curious,
see a lot of animals hanging out at.
Our depth versus high pack.
You can call a coral hanging out.
Pack
1240.
It's looking for 1, 2, 3, 5. There you are. So yeah, it's 20 meters shallow.
I think I'm pretty much lined up uphill from what my sonar shows at 255. It's hard to say, but
Are you suggesting a move?
not quite yet, but in the near future.
So we're slowly proceeding along this
Yeah, I'm ready for a move now.
pinnacle along
He's moving.
these
He's on
areas
move.
with these.
250,
we're flattening out to this
Yeah, 260 works.
still heavily
I'd go 30.
cobbled
Looks like we're
area.
gonna do.
Bridge.
Current flow
now.
looks like it's,
Another move, please. 30 meters.
or maybe
bearing
not the current flow, but we're not
260,
seeing as many
30
animals,
decimal
perhaps
two.
related to the lack of current flow.
Good copy.
Or possibly because these rocks all seem more sedimented than the others.
I think you
So
might
one
be
of the
targeting
important
me now.
aspects
Oh,
of
sorry.
That's all right.
what we study in the deep sea is what controls the distribution of different animals. Sometimes
Thank you,
you
Rich.
run into a pack of them, a lot of them in a sort of very dense aggregation, and then other times they just peter out and you don't see anything for quite a long time, or at least nothing sort of large or sessile in suspension feeding. Even though you don't see anything, there are
Lots of lasers?
invariably very small forms living amongst the rocks and in the sediment. Lots of tiny worms, lots of brittle stars living
I think they're
amongst
up
the
there
sediments.
Yeah.
Even
They're on.
single-cell
Might be
organisms,
hitting your drawer right now
amoebas
if your
living
rotator's
amongst
too low.
the rocks and so forth.
I see him.
I puppied off into a great expanse of nothingness. Find something interesting on the other side, but
Go, no one's gone
I
before.
got a tug on my leash there, so I've come back and we will linger
Okay.
until
Proceed as you can. Dave, I know you're as anxious to see things as we are.
Starboard lower. Stow it a little bit. Here we go. We got a 30-meter move in.
Yes.
Good. On the move. Sweet.
Do you see that round structure sort of in the background there?
background.
To the right, there's that triangular white thing, and then there's
Yes, I do.
yeah. I'm not sure if that's a sea urchin or possibly another sponge. Or conceivably a rock.
I'm thinking coral base, but then right under the lasers is something spongy looking. So we're good to zoom. We're going to get this triangular thing just to
Yeah, I think
satisfy
that's
our curiosity.
yeah, that's a good point. It actually might be a barnacle. Oh, we got a little hermit crab in there.
Ah, bonus.
Yeah, I think that base might be just another one of those dead coral bases. Doesn't have the plates that I would associate with a barnacle. But yeah, let's take a quick look at that hermit crab as well. Oh, wow. Hermit crabs are, of course, crustaceans. Their habitat is to pick up little shells left over from other mollusks, and they use these to as their sort of defense in lieu of sort of well-developed armor that other crustaceans have. Let's also take a look over at these branching structures. I think they're hydroids. These very small.
I think these may be a whole lot of little carnivorous sponges, actually.
Oh, really?
They have little branchlets going all the way down to the base.
Oh, yeah. Yeah.
I mean, they're a lot more flexible than I would expect for a sponge, but that's otherwise what it reminds me of.
Yeah. Oh, that's interesting. Those. Yeah. So these might all be small carnivorous sponges. Can we zoom over a bit to the?
Yeah. Now that I'm getting a closer look. Sure are waving a lot.
Yeah. I think there's a better patch of them over to the right.
Okay, let's come out a little bit.
Yeah, on the rock face there, just to the right of the hermit crab.
Actually, let me tilt down, because if they're right in front of me, we can get even close.
Like sponge more than hydroid, because I don't see any concentration of tentacles up around the top that you would expect from a hydroid.
Okay.
Here, I'm looking.
Yeah, they're.
We may only see them in silhouette.
Yeah.
But I'm going to.
Co-pilot, what was the question? Yeah. Yeah. Oh. Yeah, we meant to leave that out. What's he going to use?
Or we're going to cut into that dead section.
Thank you, Bridge. Okay. We could just use Aqua Shield.
There we go. Chris, is that...
Yeah. I don't think that's
one
the
of
same
them?
thing.
Okay.
We're looking for a bunch of sort of they're stalks, but they have projections coming off of them.
Oh, right.
And there's a bunch of, they're all about the same height.
Yeah.
And they're very, so
those
that's them.
guys.
Ah, there we go. Yeah.
Okay.
Yeah. That's exactly what, yeah.
And they are some distance away, which we could get a little closer.
No, I
if
think,
need be.
I think this is fine. I think this gives us a general notion of what they look like, but I think we just wanted to sort of get a general idea of what they look like all the way down to the base, and I think that's fine. Yeah, we can. I think we can move on.
Here,
yeah.
I think there was something else.
And then there was that
spongy
white thing. Yeah.
That move's complete, and I'm just swinging pilot, but
I'm
looking down
those
at you.
were hydroids,
85.
at least the ones that I know that have the little tentacles along
That's
the
good.
column.
Yeah, there's the thing.
Tentacles would have knobs at the tip.
back in.
Yeah. I think you're right. They seem more like
That's the one
sponges.
for sponges.
Yeah. Oh, this is interesting. Possibly a dead sponge.
Now we can get a little more zoom on it.
So, this I think is a glass sponge, and for those who have already seen, who may or may not have already seen the large diversity of glass sponges that we've shot on video. This is an animal, but its skeleton is made out of silicon dioxide. It's literally glass, and it looks like this one's partially dead because usually there's a tissue that's present around it. You can see the white areas that are presumably the living part, and then this sort of brown gray area, which is more dead tissue. Interesting. All right, we can move on. But that was a great example of how you can tune in to the fine detail in some of these rocks and see all sorts of things happening. Wow.
So off into Boringville, but
oh, come now. It's not that boring.
I'm going to get down in the sediment because I used to think there was nothing here, but there's lots here. It's just tiny.
Yes, exactly.
So we'll get down there and I'll push out ahead, buy us some time, and we'll go play in the mud.
What does your sonar suggest as uphill now? Closer to 270 maybe?
I couldn't say.
Somewhere in there.
So this is Okeanos Explorer, and we are currently engaged in the Laulima Okamawana expedition. We're studying the Johnston Atoll region of the Pacific Islands Marine Monument. We're surveying an area at a depth of about 1,200 meters. This is got a very nice basalt bottom. Lots of for a few minutes ago, we'd seen a number of corals, saw some sponges, little hermit crabs. There are shrimps all over the place. As we look on more, I think we'll find there's quite a bit of life. Just have to look for it.
Yeah, keep us moving.
Okay,
I'm just going to look into the sediment while the ship catches up.
Let's go 270.
Okay.
Let's max zoom, please.
I'm fine with 30 meters. Sounds like you want to keep going here.
There's a tiny sponge.
Yeah, so one of the things that you'll notice in the screen, there are these tiny little white domes that are on the surface of this rock.
30 meters.
These might be
270. Speed
they're very small adult sponges, or possibly juvenile sponges that are slowly growing on the substrate, and possibly that same species as the sort of round one we just saw, or conceivably the same kind of spherical sponge that we saw a few minutes ago.
Thank you, Bridge.
Let's go see what's on that rock that was up ahead. I think there was a shrimp or something else on it as well.
You're just bored with tiny stuff, okay?
Yeah, I am actually. Sorry.
Right. No worries.
Oh, did you?
It's come a little wide, please, Bob.
Right there in the center.
Okay.
Oh, you mean to the left? No, that's... I think these are all hemicorallium. Yeah. These are all the things that... What's that white thing right in the center, though?
Let's go for a video. Off-centering.
I think you're looking down on the base of a broken.
Oh, is
let's
it
go
another
back
base? Yeah. Yeah, I think that's the base of another, of something. Yeah, of a coral or something. Very well.
Just come out a
Let's
little
move on.
bit.
Yeah.
Shrimp, and then coral, and dead coral, sponge.
We are on the move, pilot. Early into a move, and I got you at 60, so
Okay.
you don't. Another minute here, and I'd like you to push out, see what we got.
Come wide, please, Bob.
This was sonar return. I think what it is it comes out, levels off, or depression, and then comes back up. But
Couple of bumps. Yeah.
I just wanted to verify that.
I'll pop up and have a look.
Thanks. Should be a non-issue. Yeah, okay. I'm comfortable.
So I'd be content to have either of you come in and fly for a while. I need a little bio break myself.
Okay.
I did this morning.
Okay, Levi, you're the man. Just if you can get
Yeah.
somebody to relieve you.
So it sounds like we have some pilot change happening soon. So
Yeah. Dry lab now.
looks like we're coming in on this. I think this is
Workshop nav.
Narella. It's a coral that we've seen earlier today with
Clear to zoom.
an ophiuroid wrapped around the inside of it. This is a common relationship amongst these corals.
Hey, Chris. If you can find
looks like
someone
the
to
disc
come up
is
for a
kind
few
of
minutes.
swollen there.
Co-pilot needs a break.
Excuse me. These have
Thank you.
unusual. These are brittle stars. These are related to
We got
the
Carter
ones
coming
that
up.
we've seen other places, but the
Co-pilot
the
got
bodies are different in that they have sort of fleshy coverings over them that provide them with a level of flexibility that the other brittle stars don't typically have. And so they're often very tightly wrapped around these coral hosts. When we get these specimens from collections, they're actually almost impossible to pry off of these corals. So they're often the implication is that they're very tightly
Just come out
associated
halfway,
with each other.
please, video. I'm going to come around the side and image the mouth. Oh, I missed that Z-bias. It's
Yeah, it's nice
so
to
for the benefit of the other pilots listening in both air and underwater,
Thank you,
the
Rich.
difficulty we're having today is we've lost our vertical trim control, and which we use to offset our normal buoyancy, and so it makes control a little bit more difficult. But Josh is working on it, and I'm sure it'll be fixed soon. Okay, let's come back in video. Just trying to come right a little. Hold
Is
that.
that move complete? Yeah, is that what I heard?
Yes, it is.
Okay, thanks.
So let's see. I guess. You want to get make another?
Oh, I got
Oh,
pretty
that's
good
a very
return in
nice
front of
view
me.
of the oral surface of these
We'll check that out first.
snake stars.
Yes, you do.
I think you actually caught a larger sponge, one of those round sphere-shaped ones over to the left there as well.
Okay, yeah. created a little bit of a snowstorm here. I'm going to hand off to my colleague Levi here. Come wide, please, video.
Oh, looks like there's a nice tree-shaped sponge over to the right, and wow, we're back on the wall.
Yeah, co-pilot. This looks like a pretty serious wall.
Okay.
None of this is apparent to the base map. We stopped about.
So,
3
there's
minutes
a little.
ago, we should probably still
Is
swing
that a
Copy.
ctenophore or
Just gonna
a
keep coming up.
amphipod and a cell?
Video, frame it up. Kill lasers.
Lasers. Off.
Zoom at will video. I'll do my best.
Lovely. So, one of the pleasures of working in the deep sea is looking upon a lot of swimming pelagic animals, and this one is a comb jelly. These are relatives of proper jellyfish, and what you're seeing is the lights reflecting off of swimming paddles that they're using to move through the water.
If you like.
These are predatory. They usually extend tentacles with cells, or not stinging cells, but adhesive cells that they use to capture food. They take on a variety of forms, especially when we're doing transects through the midwater.
Down by Z-Bias. Manual Z-Bias.
Manual Z-Bias.
All right, we'll let this guy go. Video.
Yeah, we're done. We can move on when
Let's
you've
go
got a
full
chance.
wide. Acquire this hard
Chris, I was just
sonar
going to say
material.
that the dark red pigmented structure in the center there would be the gut,
Oh,
part of the gut, and I wonder if it's pigmented to hide any possible bioluminescent prey that it captures. And then those two bright white spots, I believe, are the tentacles
anything that
retracted
does
and
involve
held
space
inside the
carrying
tentacle sheath.
oh, great. Thank you, Scott. So did we just have a front row change?
Yeah, we had a quick pilot change.
We haven't had a chance to hear from you guys today, so who do we have working at the front today?
In the front row, we got Levi Unima sitting pilot. To my right, we have,
Chris Ritter, I'm co-piloting.
and to the pilot's left, sitting nav.
Dave Casagrande, navigator.
And Bob Knott sitting in the corner. I was on the XRV.
What's this?
Yeah.
Oh wait,
Oh,
this is okay. This is another flytrap anemone, I think. Yeah, we can move on. I think we actually saw this one earlier. Actually,
We will keep moving.
yes. Another one of those sponges that Chris likes so much. I better go get some imagery of it. So there's that kind of
I
anastomosing
suppose.
bush type thing that's over to the left there.
Oh, yeah.
It's white with sort of dead branches.
It's the fourth one we've seen in this particular dive.
Oh, he's back. So, USBL isn't doing great.
And
Probably related to the cliff, but
just
Yeah,
for a heads up, I
yeah,
think there was also a lot of there was a small patch of white pods all the way down on the lower
Okay,
side,
video,
but
let's zoom.
I'm not sure if we're going that way. So this
Are there any
is
sea stars?
nope. So this is Chris Kelly back in his chair and looking at this sponge that he's been obsessed with since we saw it. It's a new species, and I'm going to go get lunch, and I'll let him talk about it.
Yeah, it's about time because now we're going to see a sea star finally.
Rotator coming down.
And trying to get.
The sad thing is, you're probably right.
Yeah, we can do a snap zoom on this since we got some good video of it already.
Okay. All right, video. It sounds like we got this.
And what we're just making sure is all of these have got this weird cnidarian. That looks like the little spike sticking out of it. And sure enough, this thing is covered with these zoanthids or hydrozoans or anemones. We're not really sure. You can see their tentacles waving right now in the current. So these are not stiff parts of the sponge. They're actually a commensal cnidarian that is living presumably peacefully with the sponge because the tissue's live where the cnidarians are coming out of it.
Yeah.
So this is not that common at all on sponges where you actually find this amazing association. So it's very, very cool. We think this is a brand new species. I've been getting some feedback after sending pictures to some of the world's authorities. Dr. Konstantin Tabachnik there at the Shirshov Institute in Moscow. Dr. Henry Rijswijk in University of Victoria. Rob van Soest in the Netherlands. So we're all pretty excited about this thing.
Just give you Chris Kelly an idea of what we got here.
Well, a nice little stand of these deepwater precious corals, corallium, hemicorallium, sorry. Tina Molitsova has joined us. Maybe Chris Ma already called her out. So glad to see you, Tina. She's also from the Shirshov Institute in Moscow. Les Watling is with us, the University of Hawaii. Katie Musser and Bruce Bundy, his premier fish biologist, just got back from vacation. So we're looking forward to hearing his voice on some of the dives that we're going to be doing on this cruise. Sako Matsumoto from the Chiba Institute in Japan, she's with us as well. So this is great. We've got some experts on the line to help out with this.
Okay, watch this. We have a pretty hard return to Sirius's port. We're going to check it out real quick, and then we'll back to the bottom here.
Roger that. Thank you.
Slight detour. What do you think?
Oh, right.
Co-pilot.
Hello, pilot.
Back you out, see if we can see it on your sonar.
Shoot on around it. Pilot, on video, I mean, you gain up to look into the darkness. Yeah. Okay, so 90 degrees off.
Give it a little tilt up. I can maybe give you some more.
Yeah, absolutely. Okay.
So has there been anything collected so far, pilot, while I was at lunch?
Nothing while you were at lunch, no.
Okay.
Back down 30.
Well, we're always on the lookout for another rock. These are clearly pillow lavas. If you just see something that has a chance of being loose, maybe we ought to
head towards it, Nav?
maybe we'll grab our second rock here.
Go either way.
Is that a walking crab on the bottom?
Looks like a lot of this is consolidated, but you never know.
The next waypoint is 235 from us, so I think we should head that way. We were following.
Did you guys?
Let's turn to port video. I heard you. We're deciding the next direction here. Well, actually, just something you'd like to see, immediate view? Maybe
There's something crawling down to the lower right. And also, can we get the lasers on at some point?
Yeah.
Great, thank you.
Can you grab lasers when you get a second? Video, frame up.
That is a crab carrying an enemy.
He's got his partner in crime there.
I don't think we've seen these so far today, unless it was while I was at lunch. This is a
Come in video at will.
decorator crab in the family Homolidae, and we believe this is Lamoha longirostris. And you can see he's carrying an anemone in his back claws there, and that's what they do. Every time we see this crab, they're carrying this anemone behind them. So he's got two claws right in the hind end that he's not using for walking, and that's what he's using to hold up that anemone. So obviously they both get something out of this relationship. The crab getting some protection for his rear end, and the anemone getting to move all over the place and maybe do a little more efficient filtering.
Do my best here.
But Levi's such an ace that makes it look easy.
Max zoom.
Max zoom. So yeah, co-pilot, I'm thinking we'll line up with that sonar return and acquire and go up, and that's
Video's
also
clear.
That's the right.
That's the right direction.
We have
So
to
follow you.
veer off this way.
okay, video. Let's come wide.
Thank you very much.
Let's
Get your tilt back.
get my tilt back.
Look at these pillows. Jeez. All right, there is no mistaking that this is volcanic seafloor that we're looking at right now. Beautiful toothpaste-like tubes of lava. So you're actually seeing frozen in time an event that took place millions of years ago. So the only difference is back then when these tubes were formed, they were molten hot and creating all types of sort of clouds and disturbances and everything else as they came out. And now they're kind of cold and lying there exactly the way they looked, with the exception of some crust over them and maybe a little bit of erosion. You're seeing something, you're getting a view at millions of years ago, what the seafloor looked like. Yeah, there's a little something right on that rock, pilot.
Possible stock sponge.
Yeah, I would agree.
Going for it now.
Pilot thinks it's a stock sponge.
Range of that return about 20 meters.
And after this, pilot, there's a bigger coral behind it that we maybe if we could get a look at if we're heading
Sounds
that
great.
way.
We can do that. We'll get
You
nice and close.
preempted my request.
I'll get
I
closer
think
to
it's
it.
different, Scott, and I think that's why you were going to ask for it too. But we'll find out after we get to sponge,
Okay,
which
but you
is
can
different.
frame it up as I come in for a landing.
The ball on the sponge is about
Okay.
6
We
or
can check
7
all the zoom we want
centimeters.
Video. Oh, actually hold.
Bolosoma, that's
Okay.
right.
Yeah, now you can zoom video. I'm more
Can we get
stable
it?
now. It's a little bit of a precarious perch
It's a
here.
sponge we call Bolosoma. It's a stalked glass sponge in the family
Yeah, if you could rotate up to
Euplactelidae.
the bulb of it.
Taking your tilt.
Very
Should be a
distinctive
slight
appearance. It's an easy one to identify. It's got that great big osculum or opening right on the top of the ball there, and then the other face is kind of scooped out, and a lot of them look this way, and it's so distinctive that we can actually get at least the genus
Video's
on
clear.
this
Video,
particular
yeah. Come
sponge.
wide. I think the opening's on the other side. I think.
Yeah. So we can look at that
Yeah.
coral, that big fan.
Copy.
Come on, Scott, tell us what it is before we get too close.
Just my swing arm on
Well,
that
certainly
rock on my
looks
starboard.
like it's going to be another
Yeah,
primnoid.
awesome.
And what are we looking at
Should
here?
be
Maybe
Co-pilot, if you're comfortable, I'd like to
always
cap
forgetting this one
Sirius
has
and
these
check its
swooping
black balance.
branches.
It looks green.
Yeah, no problem.
Yep, I think those are your two possibilities. The one you mentioned, Calyptrophora or Paracalyptrophora. It could be Calyptrophora angularis, but I'm not really sure. It could also easily be in the genus Paracalyptrophora. Right.
I think we both agree that it's a member of the family Primnoidae.
A little closer
We can
video.
certainly go that far safely.
Okay, there we are.
Yep, I agree.
Okay, video, you can frame this up on D2. Yeah.
Two crinoids.
I'm just
at the
going to log
top,
it as
and anemone in the
Calyptrophora
middle
or
top.
Okay, I'm gonna turn
Paracalyptrophora
the
just to
starboard.
narrow it down a little bit.
Co-pilot,
distinguish
second,
it from
you can
the
rotate me up.
Norella that we've been seeing all day.
Thank you.
Hold on the anemone if you can.
Anemone, going for anemone here.
You got a an enemy growing on one of the branches.
Brittle star was just to the right of the anemone.
Almost looks like the polyps
Coming to
are
the right, easy.
under the anemone. Which are the I don't know what's going on there. Those little bumps in the back of it.
Yeah, I see what you mean. I wonder if those are
Holding here.
former little new branches that were arising that got covered up.
Yeah, it could be.
You'll go up the right-hand side.
Copy. Co-pilot, rotate at will.
Oh, there's a crinoid.
Track.
It's a feather star right there to the right of the screen. This could be something in the family
Fish
Antedonidae.
gonna float by.
Oh, there's a fish.
We could ditch this if you'd like to see
Yeah,
the fish.
please, let's ditch it and get the fish.
From
We got
wide
some
video.
good.
Where'd it go?
There he is. What do
Frame
we
it up
got
video.
here?
I'll do my best.
I still got you tilt.
Tilt way down.
Copy. Coming down.
Aha. Okay.
That may not be a fish, Chris. Oh, no, it is. I'm sorry. I thought it was a squid for a second. That's another one of
Good,
the halosaurs, the
Try to
deep
lead
sea
it.
spiny
He's coming
eels, the
back to your
halosaur.
left.
If we can
He's
zoom in on
exactly
the
that
head,
full.
seeing the top of the head,
Pulling
to
out
tell us if
wide.
it has scales over the snout or not. That would make it Aldrovandia. It looks like Aldrovandia from the general shape and the color. So these are pretty abundant worldwide. They're distantly related to the true eels, but this has a fin on the first third of the back, a dorsal fin sticking up that the true eels don't have. Long whip-like body with an extended sensory system along the body. You don't see scales on the top of the head, so that would make it Aldrovandia, and the species is probably Aldrovandia phylacra from the color pattern. You can see the brain exposed in a very thin skull on top of the head. That's the kind of yellowish color on there. I'm not quite sure what that's for. These animals live below the depth at which sunlight penetrates, so it doesn't have light sensing function. So it's a halosaur, Aldrovandia phylacra.
Thank you very much. That was the voice of Bruce Mundy, who is a premier fish biologist, and we are very delighted he is back from his vacation, although he may not be.
We'll let it go.
But
There's a cool rock right here anyway.
we've certainly missed you for a bunch of dives, Bruce, and it's great to hear your voice. And
Thanks, co-pilot.
he is our fish authority here for these dives, because he knows a lot of these animals. So we're now getting a little bit of a large block, and you see this darker red coral that's coming up to the center, upper center?
Upper center.
It looks like a little line, but a brighter red line.
I think that's a shrimp.
Oh, yeah.
Oh, is it a shrimp?
Yeah, dark red.
Oh, it is a
snap.
shrimp. Sorry, I thought it was a coral on edge.
Yeah.
Okay, it's a different one. So this is Heterocarpus.
Getting closer video.
Copy.
Approaching.
And
Tracking.
it looks like it's something that's similar to Heterocarpus levigatus. I thought that was possibly a Paragorgia on end, but only missed
Pretty good sonar returns, huh?
the ID by a few
Center
phyla.
up now. Getting close.
Shape and color were right.
You still need a closer look. Watch these.
Okay, thank you.
Yeah, just keep what you got there. It's a good
Yep,
shot.
I can see features of this particular shrimp as well, the serrated rostrum. You can see the spines on it. It's got a keel that's running right along its thorax there. So I'm fairly comfortable with Heterocarpus for sure, and probably Heterocarpus levigatus.
One more center of his head. I'll max out the zoom.
Sorry, video.
Yeah, look at that. What an amazing detail.
That's it, video. Let it go.
Great, thank you very
That's
much.
an excellent shot.
You know, it wasn't a coral, at least it was a new observation for the day.
There's that wall.
There it is. Serious view, you can see it.
I knew it was going to be something like that.
This is quite the wall feature we got here.
Yeah, nice.
It'd be good to rotate side to
another
side.
wall of beautiful corals,
Come
well,
yeah,
most of which are in the family Coralliidae.
turn to the right to get a sense of what we got here.
We're not seeing huge colonies. We're seeing a lot of smaller colonies and I think some moderate-sized colonies, but would suggest that this particular stand isn't that old. There's something right, right.
Snap on this. Let's see if we need a closer look at this guy, or if this is.
That's okay. That's one of those crabs that's carrying the anemone.
Okay.
Thank you.
Yep. And then this is what we got to the left here. What is that big thing?
It's like a very fat starfish on the back of a sponge.
Is it where?
The white thing.
Oh, you got to zoom in on this. Chris is gone. Chris is gone, and we see a starfish.
We knew this was going to happen.
And this is a good angle because this is showing the other
Unbelievable.
side.
Every time he steps
So
out.
keep this
I
angle.
don't think it's going to be.
It isn't? Am I hoping too much for it to be a starfish?
Roland called
You
the
snap
starfish.
video as I approach.
Yeah, it's a starfish.
It's a starfish.
This is a
Don't let him.
sizable subject here.
It's a big one.
Just hold that.
It
Don't
is.
continue to
It
turn.
is
We're looking
eating
at the other
this
side.
coral.
Okay,
Can you
laser's
hold
coming
on
off.
for a second,
go back.
pilot? Or are you really stuck?
No, we're okay here.
I'm actually going to run downstairs, see if I can get Chris in case there's.
You can have the navigator call the.
Yeah, I'll call him.
Yeah, please do, because
Can you rotate back and
yeah,
look
I don't
back
know
at
if
its
this is
underside?
something that he
Like, you know, the
he might be
emails you
interested
Should be
in.
on the list there. There's a fish to my right. I could. He's way. Never mind. The fish is too far away. I see it way up. Sorry. Video, come on. Zoom out to keep him in view here, just so I know where he is.
Watch your upper swing arms.
You're full wide.
Thanks, video.
Say,
Sorry,
Yeah.
we're still chuckling. This is because Chris
I
Maughan,
gotta
my
stay
co-lead
relatively
here, is a
perpendicular.
sea star expert, and
Well, I
whenever
think
he
my
steps
minis
out
do
for
so I
lunch, we see a
Look
sea star.
up to my light bars, see where that brow is.
This, I don't know if you heard me say, I was saying that this just does not look like a fair fight. This is what looks like a pretty large asteroid relative
Okay,
to the
video,
coral.
frame
I
it
don't
up.
know that I've
He's not in the mess. He
seen
might already be
kind
on
of
his way
size
back.
disparity.
Right, I would agree.
tilt down.
This species maybe, boy, Chris needs to be here.
I can
Can't
make a
even
page.
really have. It could be Hipposterius. It could be some other
It's gotta be.
genus
Yeah, I
of
can-
sea star that's predatory on corals.
Can you fully
And
deploy
Scott
my uppers
France,
so
that
they
was
put
his
back a
voice that
little
you
bit?
just heard from the University of University of Louisiana at Lafayette, was just remarking that this is a huge sea star for this size of coral. We don't often see this.
Roll an active page.
Fully deploy.
You can also say that it's
Just
not
so they're,
currently
you know,
feeding.
further
You
back.
can see its mouth in the center there of the radiating arms, that sort of darker space right in the center, and
Right,
it's
they're
not
probably pretty
oppressed
close.
right now to any of the branches. And I believe this is probably one Crystal pointed out
I'll
that
try
would
to get
invert
used
its
to your
stomach
shot
upon
video.
feeding, and that is not going on
I'm floating
at
right
present.
now.
You can see some of the tube feet tucked in the grooves there, and they're all kind of meeting together in the center,
But how about
which is
a
where
page?
the mouth would be.
You could get a toe in there, you know.
Well, I think my brow might hit before my toe, but we'll get
Well,
it.
you can use your brow.
That's what I'm thinking. Just wanted to get that mini Zeus on it.
Okay. In
I'm
that
slowly
case,
approaching.
So, pilot, is it possible just to sit here for just a
Yeah,
couple minutes?
yeah.
I'm going to run downstairs, see if I can get him.
Kelly Elliott and others have gone to find him, but not in the channel.
I called the Mass Week Occasion.
People are looking for him, but Kelly.
Okay.
Okay, just hit this way.
And once you get your forehead up there, I'll see what I can do with the lower swing arm.
Copy. It's got to be close, right? I'm right.
Come on, Chris.
Estimate you be about an inch away.
I just feel the bump.
Is he
Lower
coming?
swing arm
Oh, he
coming
is.
in.
Okay, so Chris Ma is on
There
his
he
way.
is.
There
He's
you
wearing a
are.
starfish
So
shirt.
we just. So I wonder if we could zoom in again for Chris.
Yeah, absolutely. I'm trying to get a
Yeah,
steady
we're just
shot.
trying to get the vehicle stabilized. We haven't yet got the first best shot yet.
Yeah, video. Frame it
It's
up.
good lighting there now.
Frame it up, video.
You're still bouncing up.
Yeah.
Yeah, this is looks very much like Hipposteria muscipula, which is a species I described a few years ago.
You already have it.
Predatory
Trying to get
on
you.
lighting is good.
bamboo corals
Change
before.
anymore.
Looks like they might actually
Tilt the camera
now
up.
feed on hexacorallium.
Bye.
There's a shrimp.
You don't really see a stomach. Yeah, it is. I guess it is. Is that
Oh, yeah, they're-
inverted? Or
Well, it might not be feeding. It could just be sort of. It might have already fed and will continue to feed later on. Because from what I've observed, and from what I think Scott told us before, sometimes they'll just climb on and park there until it's time to eat again. There's
More
probably see some
a
of the
little bit,
damage.
not much,
I
no.
think. See some of this area where all of the tissue was worn off. I bet
I
that's
see.
that's post starfish eating damage.
Starboard, upper,
Yeah.
Starboard, lower's okay. I don't
And then
know
you have the live
starboard,
polyps
upper.
over on the right side, which
just
haven't
turned
been
it down.
Yeah.
consumed yet.
Yeah.
I see.
But yeah, as much as I'm tempted to,
You
this
don't
is
have any
something
more
that
rotate left.
No, roll. No.
We've,
upper swing arm
we've
board,
seen
rock,
before.
I think.
Okay. Well,
Some nice views
Pull
of the
that
pedestal
lower
area,
back
though.
because it's not doing any
There's
good.
our claw-shaped structures that are on the underside. I've never been able to figure out what they use them for, other than
Actually,
perhaps
that
to
was providing one for light.
as defense against small predators.
The lower?
See these
Yeah,
little
the lower.
sort of
Whatever you changed got
little
worse.
clam-shaped
Okay,
or
bring it
lip-shaped
back.
things that are on the underside. I've always wondered if maybe they had a
Cold.
role in.
Got
Oh,
it.
thank you. That's delightful. The light helps create quite a lot. But I've always wondered if maybe these are open, and so I was wondering if maybe they aid in feeding somehow.
you're talking about these things, right?
Yeah, these little
Yeah, I
there's
see
these
them.
little clam-shaped
They look
things.
like little wings or something.
Yeah, they're like an open cosmetics
Sorry,
case or
wrong
something.
tilt.
Yeah, see there.
Yeah.
Um,
Oh, you see a lot of them right in the center here.
yeah, and they're all open. That's interesting. I bet they help with feeding
Starboard, upper,
somehow.
like
Slight
close
tilt
to it.
up.
See, this
Boy,
is.
that's amazing
Stand by.
detail.
Oh, no kidding. Those things are
Tilting up.
only about a millimeter across, or a centimeter across. So,
Got it.
Yeah.
I can hear you, Dave.
but in a lot of
Pressing on
ways,
it.
this would
I was
actually
wondering if that's
this
full
is
or if we can do a microscope
just as
view.
valuable as
That is
discovering a new
excellent.
species
Okay,
is
excellent.
discovering what some of these structures do, because these have been observed on sea stars since they were described. In some cases, the 1700s or so, and no one really knows what they
Pulling
do
out for
exactly.
slope.
So actually observing
Full.
them would be very very interesting. Still not quite sure what they're actually
The next
doing,
shot
other
will
than the
be
fact that they're all
the
open,
that arm in the lower
feeding on
left.
this animal. What do the lasers? How big is this?
Let's come out a little bit more for
It's
If we
a-
could get the lasers.
It's a big boy.
Get lasers when. Yeah. There you go.
There's one. Oh, there they are. So it's right.
Yeah.
1, 2. There you go.
So it's 10. Wow.
15.
Yeah,
well,
so
you're
it's
seeing
about-
it at an angle
yeah,
too, so
it's
maybe
about
20 centimeters.
Yeah. So it's about the typical. Yeah, that's about as big as the ones that I've seen before.
Okay, can we see the lower left
Wow.
arm?
So are you gonna leave and finish your lunch so we can see another sea star?
You know, I was just telling Annie this. I
As far
was
left
like,
as I can get you
what?
rolling.
Yep.
What is it? What is it with
Steady
you
shot.
and this whole thing?
I'm just noting relationship. That's all.
Yes, and I find it extremely frustrating. This is almost comical how this happens. I should just go into the stateroom and wait there until the next one shows up.
Well, I know. I think we need some more data points. We've got 3, so we could analyze this statistically already. But
Yes, whether or not there's some kind of inverse proportional relationship with
Okay,
my
you're
being able
cleared.
to
pulling for full coral.
completely
There's
miss
a set
starfish
there.
whenever they show up on screen. Oh, there's a
Tilt
little
up.
crustacean there.
so are we good on this?
Yeah,
Chris?
no. Thank you so much, and thank you to our program administrator Kelly Elliott,
Slight tilt up.
who went and got me
Okay.
so that I could rush up here and see this. That's a beautiful animal,
Yeah.
My video back up.
and even if
Okay,
we
hold
didn't
on.
collect it, certainly the highlight of
Catch, tilt
seeing
back.
that. This is one of the species I described only a couple of years ago, so certainly
Bring
it's a
that
highlight.
Starboard, lower, back outboard.
You can see some of the other.
Guys, where's the laser
Full
sign? Do
wide.
we know how
Full
sea
on.
star is?
Thanks, video.
Yes. Go ahead, Chris.
Oh no, it was about 15 to 20 centimeters across, so quite a big one.
And then that port upper, you can fully.
So
deploy that one again.
that was Hipposteria muscipula.
Or we can still
and,
it if we think this is going to continue.
The name actually refers to
Maybe that fish was off to your
muscipula is
right.
what they call a flytrap the Venus flytrap. And the reason I called it that was because those pedicelaria have teeth on them, and if you look at them up close, the sort of pedicelaria look like Venus flytraps.
Explore to the. We saw this guy before.
Seems to be one missing. It's an enemy.
I think we imaged him.
I see some of these have damaged, or the tissue's been eroded off of some of them. I wonder if
those white ones, you guys saw
starfish
those?
has been busy.
Yeah, we got a really steep wall here. This is
Yeah, serious view gives you
Fish, very bottom right, possibly.
Oh yeah, those of you, if you look at feed 2, you get an idea of what the overall terrain looks like. It's really
you still see that?
really beautiful.
It's that squiggly thing. I can't tell if
Yeah,
it's
let's
moving.
snap on it. It might be a stock of some sort. Let's do a quick. Yeah, I think it's stationary. Yeah, it's
Yeah,
okay.
it's stuck.
Stock.
Yeah.
Let's come back out.
Yeah.
Okay.
If you rotate around that
there's
nose, that'll be a good view from
a
Sirius.
little bit bigger colony of corallium as well. So it's it's sort of a mixed community here.
Yeah,
some
we get that
larger
port
and
upper
some smaller
all the way
ones.
deployed. I think it's kind of partially. So
Okay,
it's
port upper.
hanging out.
Deployed.
This is actually a really good find. This is a moderate, potentially evolving into a high-density stand here of these
Yellow coral
corallids.
top right.
Yeah, cool.
Oh, what's that yellow one up on the top there? Looks a little different.
It looks like it's with zoanthus, but yeah, you're right. I don't think we documented. If we could get that yellow on pilot
These
right
look
up
like
there to
little
the upper right.
chimneys. Okay, coming up.
I don't think I'm overhung, am I?
No.
Okay. All right.
Oh, yeah, look at this. How about the broadside
Right,
one that's
right.
over to the left, because
Yeah.
you have a better view of that?
Might lateral left. Get yourself.
Oh, this is turning out to be a really pretty community here.
Yeah.
Yeah, we did. Roland just pointed out again, those of you might want to take a look on the Sirius view too, just to get an idea of the amazing topography we're on right now.
And is that
That's a good thing.
there's a white globe thing that's in the center
Yeah, let's-
rock there when
we
we're
can get that, yeah.
I'm trying to watch
Not sure
that.
if it's an urchin or something else.
Copy. I'm setting up for the possible gold coral. I think.
Yeah, it's awfully deep for gold coral, so that's what.
Okay, Vito, let's try to zoom from here.
It's overgrowing.
Yeah, it does. They're really big.
There's your sponge again.
Yeah, there's the sponge, the Tritodictid.
I'm going to slowly get closer, video. Approaching.
So a beautiful bunch of zoanthids. They're parasitizing this corallid hemicorallium.
Sorry,
They're
yeah.
growing over it. That's the yellow polyps that are growing over the pink polyps and have almost taken over
Zoom out a
the
little bit,
entire
video, as I
colony.
set up.
And there is a squat lobster right on the zoanthids. So everybody's living on everybody else down here.
Yeah, I think we're going to do some floating. I'll do my best.
We wanted to get a quick look
yeah.
at that little.
Let's snap.
Yeah, thank you.
I think this is what they want.
It's a sponge. You are good at spotting sponges, Chris. Thank you very
It's
much.
my curse and also my power.
As I center, you can try to zoom, but it's not going to be extremely steady. Yeah.
So that sponge is a curious little thing.
Yeah, it looks like a brain.
It's some type of a euplectellid. I'm not sure what it is.
We're going to do a quick pilot change up here.
Wow, these squat lobsters are enormous.
The laser's still on.
Negative.
This is really pretty view right here with these beautiful, colorful corals. You know, on this really steep topography. But I don't know if this is possible to do a snap zoom to that little yellow string that's down on the lower right third.
Josh, you can talk to Josh.
Oh,
about
it's
that.
Josh.
We're going
Sure.
to swap out for 1 second.
Got it. Okay. We're going to do a pilot change for those of you watching, so it'll be a moment. Probably just zoanthids